Good evening, everyone. I welcome you all to the week 8 live session for the Bioinformatics and Algorithms course. Yeah, so in this, as usual, I'll be discussing the summary of the week 8 lectures. Then I'll be proceeding towards some sample problems which helps you to take up the assignments. Then I'll be taking questions from the portal as well as questions from students. Okay, to start with, okay, this week lectures mainly discusses about protein folding and others related. So starting with the first lecture of this week, protein folding is the process by which protein chain acquires its native functional and 3D structure. So this 3D structure of a protein determines the function, and therefore protein folding, which allows protein to acquire this 3D structure, is a critical step in biological process. In bioinformatics, the study of protein folding involves the prediction of protein's 3D structure from its amino acid sequence. The process of protein folding is complex and involves variety of factors including amino acid sequence, physiochemical properties of amino acids, and the environment in which protein is folding. For example, the globular proteins have an environment of water, whereas membrane proteins have environment of lipid. So generally the protein folding process can be divided into three stages, like primary, secondary and tertiary structure formation. So in the primary structure formation, as we all know, the primary structure of protein is determined by its amino acid sequence. Amino acids are linked together through peptide bonds to form a linear polypeptide chain. In this stage, the polypeptide chain is synthesized by the ribosome during translation. Second is the secondary structure formation. The secondary structure of a protein refers to the regular repeating pattern of hydrogen bond between the amino acid residues in the polypeptide chain. The two most common secondary structures are alpha helices and beta sheets. The formation of secondary structures is driven by the hydrogen bonding between the backbone atoms of polypeptide chain. Next is the tertiary structure formation. So the tertiary structure of a protein is the final folded 3D structure of the protein. The folding process is driven by a combination of hydrophobic interactions along with van der Waals and electrostatic interactions. The hydrophobic effect is the most significant driving force for protein folding, as it results in protein folding into a compact and energetically favorable conformation. In addition, disulfide bonds can also form between cysteine residues, stabilizing the protein structure. In recent years, there has been significant progress in the development of computational methods for predicting a protein structure from an amino acid sequence. This method uses a wide variety of approaches, including homology modeling, ab initio modeling, and machine learning methods, where uh, homology modeling is a technique that relies on availability of 3D structure of a protein of similar 3D structure of similar protein, and ab initio modeling, on the other hand, involves predicting the 3D structure of protein based on its amino acid sequence and physical properties. Overall, protein folding is a complex process that involves a variety of factors including amino acid sequence, physiochemical properties, and environment. Understanding the process of protein folding is critical for understanding protein function and developing new therapeutics. In bioinformatics, predicting the 3D structure of a protein is a key challenge uh, that has a significant implications for drug discovery and personalized medicine. So here I have a set of questions. You can answer these questions using the chat box. Just a second. I think now everybody can see the questions. Yeah, here I'm showing you a few questions. You can answer the questions in the way they appear. The answers can be provided using the chat box option. So, for those who joined late, we are dealing with the week 8 live lecture. So, here this lecture will focus on how protein folding and protein folding rate, and then followed by we'll be discussing about protein interactions, various types of protein interactions. So, here I have some questions, basic questions that we have discussed in the previous class that are associated with the different levels of protein structure, where you can provide answers using the chat box. If you are having any doubt, you can ask through chat box as well as you can unmute yourself and ask. I'll wait for a minute to get the answers. Okay, no one yet. Okay, here the first question is what structure of a protein refers to the local folding of a polypeptide chain? So we know we have four levels of protein structures. So which structure refers to the local folding of a polypeptide chain? No answers on this. Primary. Yeah, okay, I'll come to the answer. Any other answers apart from primary? Okay. Here, secondary structure of a protein refers to the local folding of polypeptide chains. Okay, moving on to the next one. Which structure of a protein refers to the 3D arrangement of its secondary structural elements? Okay, yeah, no issues. Even if you are not sure about the answer, you can give it a try because we'll be discussing the answers here. Yeah, I got an answer. I request everybody to provide the answers so that will be useful for our discussion. Yes, the 3D arrangement or the arrangement of secondary structure elements stands for the tertiary structure. Next is the what structure of a protein refers to the association of multiple protein subunits to form a functional complex. Okay, I hope I'm audible to everyone. If someone is facing the issues, okay, I'll put a message as well. Yes, this is the quaternary structure includes which refers to the association of multiple protein subunits. Okay, here is the fourth one. Hydrogen bond between amino acid residues are important in the formation of what structure? Okay, any other answers for the fourth one? So hydrogen bonds between the amino acid residues, yeah, it is the secondary structure. Next, yeah, okay, I got your answers. So moving forward, what structure of a protein can be disrupted by denaturants such as heat, extreme pH, or could be some chemicals as well? So what structure could be denatured or disrupted using denaturation, chemical denaturation methods or thermal denaturation methods? Yes, it's a tertiary structure that get disrupted by the denaturation methods. Next is the, okay, it's a simpler question. The alpha helix and the beta sheets are examples of, this is the basic one, the secondary structure. Okay, moving forward for the weak summary. So I'll be starting with the experimental techniques that are used to study the protein folding rate. So protein folding rate can be determined or studied using variety of experimental methods. So first, let me start with the circular dichroism spectroscopy. So this method uses the fact that secondary structure of a protein can be determined by measuring its ability to rotate polarized light. By shining the circularly polarized light on a protein solution and measuring the difference in absorption of left and right circularly polarized light, one can determine the protein secondary structure. The changes in CD spectrum over the time can indicate the changes in protein secondary structure during the folding process. Second is the fluorescence spectroscopy. So this method involves the measuring of fluorescence emitted by protein molecule after it has been excited by light of certain wavelength. By labeling specific amino acid residues with a fluorescent dye, one can monitor the changes in protein tertiary structure during the folding process. Next is the mass spec. So this method involves the labeling of amide hydrogen atoms in the protein with deuterium, which is heavier than hydrogen. Then by monitoring the exchange of hydrogen for deuterium over time, one can determine the folding rate of different regions in protein. Followed by this, we have NMR spectroscopy. So NMR can be used to study the protein folding kinetics by monitoring the changes in NMR spectra over time. By labeling specific amino acid residues with isotopes of nitrogen or carbon, one can monitor the changes in protein tertiary structure during the folding process. Next is the kinetic analysis. Yeah, kinetic analysis involves monitoring the rate of protein folding using various methods such as stopped flow or continuous flow techniques. By measuring the rate of change in the different structural features of the protein, one can determine the folding rate. Next is the FRET technique. A FRET stands for fluorescence resonance energy transfer. This method involves the labeling protein with donor and acceptor fluorophores and measuring the distance between them using the folding process. As protein folds, the distance between the fluorophore changes, resulting in a change in energy transfer efficiency that can be detected. Overall, these methods offer different perspectives on protein folding kinetics and can be used to complement each other in studying folding process. Okay, here we'll take up a few questions on the techniques. So starting with the first one, so what spectroscopy can be used to monitor the changes in protein secondary structure during the folding process? I repeat the question. What spectroscopy can be used to monitor the changes in protein secondary structure during the folding process? Yes. Yes, it's circular dichroism spectroscopy. Okay, moving on to the second question. Dash or what state of a protein is typically used as a starting point for folding studies? I repeat, what state of a protein is typically used as the starting point for folding studies? Yes, it's right. The unfolded state or the denatured state is used as a starting point for the unfolded study, folding studies. Oh, sorry. Okay, which method involves labeling of amino acid residues with fluorescent probes? It could be fluorescence spectroscopy, or particularly when we take using multiple things where we have different types of fluorescent probes like donor and acceptor fluorophores. So it's a FRET also. So both stands here, fluorescent spec and FRET. So what state of a protein is often used to model the system for studying folding kinetics? So what state or what kind of the reaction we consider for studying the protein folding kinetics? I gave you a hint here whether we use two-state system, single-state system, or a three-state system to represent the protein folding. Yes, we use a two-state representation, that is unfolded state to folded state transition or folded state to unfolded state transition. Okay, okay, moving forward, yeah, we'll be discussing about the folding rate constant. So it represents the rate at which protein transitions from an unfolded state to a folded state. The value of this rate constant, or shortly here I denote as Kf, depends on many factors, including the size and complexity of the protein, stability of the folded state, and presence of other molecules that may affect folding. There are several models that can be used to describe the protein folding kinetics. One commonly model is a two-state model, which assumes that protein folds directly from an unfolded state to a stable native state without passing through any intermediate states. So in this model, so that is in two-state model, the folding rate constant can be calculated as shown here, where Ku is the unfolding rate constant, delta Gf is the free energy change associated with folding, R is the gas constant, T is the temperature, and the unfolding rate constant represents the Ku, represents the rate at which protein transitions from folded state to unfolded state, and with this value can be experimentally determined. The value of free energy change delta Gf can also be determined experimentally using methods such as equilibrium unfolding studies, in which free energy change associated with the folding is calculated from the equilibrium constant of folding reaction. So we call it as a stability as well. Gibbs free energy of unfolding as well. The folding rate constant can then be calculated using the equation above. It is important to note that two-state model is a simplification and may not accurately describe the folding kinetics of all proteins. Some proteins may fold through intermediates or may have multiple stable states that can affect the folding rates. So in these cases, more complex models can be needed to accurately describe the folding. So we'll discuss about the different complex models. So as we all know, protein folding itself is a complex process that can occur through multiple pathways and involve different intermediates. Two-state and three states are two simplified models used to describe the protein folding kinetics. These models assume that proteins fold via two or three distinct states respectively. So in the two-state model, a protein transitions directly from an unfolded state to a folded state without any intermediates. And here is the equation for the folding rate which we discussed. In contrast to this, the three-state model assumes that the protein folds through one or more intermediates before reaching the final folded state. The folding rate constant in model can be described using the formula shown here, where Ki and the Kf are the rate constant for formation of intermediate and the final folding step respectively. Like Ka is the intermediate formation rate constant, Kf hyphen is the final folding step rate constant. So this equation assumes that folding occurs to two rate limiting steps, that is formation of intermediates and the formation of final folding step. The overall folding rate constant Kf is then determined by the relative rates of these two steps. In both models, the value of Kf depends on various factors such as size and complexity of the protein, stability of the folded state, and the presence of other molecules that may affect the folding. Experimental techniques such as kinetic measurements, stopped flow spectroscopy, NMR spectroscopy can use to determine the folding rate constant and understand the folding pathways of proteins. Overall, the two-state and three-state models provide simplified descriptions of the protein folding kinetics but may not accurately represent the complexity of folding in all proteins. So more complex models such as those include multiple intermediates may be needed to accurately describe the folding kinetics of some proteins. Okay, here moving forward to discuss about uh, how a broad classification of proteins like fast folding proteins and so slow folding proteins. In general, protein that fold quickly tend to have higher proportion of small and hydrophobic amino acids, while proteins that fold slowly tend to have proportion of large polar or charged amino acids. Okay, discussing them in the detail. Look, starting with the fast folding proteins. Fast folding proteins tend to have compact and stable native state, which means that the folding process is relatively fast and efficient. In general, fast folding proteins tend to have high content of hydrophobic amino acids such as alanine, leucine, and isoleucine, which tend to cluster together in protein's interior. These hydrophobic interactions helps to stabilize the protein's native state and promote rapid folding. One example of fast folding protein is Villian headpiece subdomain, that is HP35, which folds into its native state just in a few microseconds. So this contains a higher proportion of this HP35 contains a higher proportion of hydrophobic amino acids, like small hydrophobic amino acids such as alanine, valine, leucine, in which helps to stabilize its compact. Next is the slow folding proteins. On the other hand, slow folding proteins tend to have more complex folding landscape, which means that the folding process is slower and more prone to misfolding. Slow folding proteins tend to have higher content of polar and charged amino acids such as arginine, glutamate, and lysine, which can form complex electrostatic interactions that slow down the folding process. Slow folding proteins also tend to have a longer and more flexible regions, which can adopt a wide variety of conformations and make folding process a more difficult one. One example of slow folding proteins is the enzyme adenylate kinase, which takes several seconds to fold into its native state. Adenylate kinase contains a high proportion of polar and charged amino acids such as glutamine, glutamic acid, and lysine, which can form strong interactions with the water molecules and other polar molecules in the environment. These interactions can slow down the folding process by creating a barrier to the formation of protein's native state. However, it is important to note that there are many exceptions to these general tenets, and folding kinetics of a protein can be influenced by many factors, including the presence of disulfide bonds, post-translational modifications, and interactions with other molecules. Therefore, predicting the folding kinetics of a protein based solely on its amino acid sequence and is still challenging task. In summary, the preference of amino acids in fast and slow folding proteins is generally related to their size, hydrophobicity, and polarity. Fast folding proteins tend to have a higher proportion of small and hydrophobic, while slow have higher proportion of larger hydrophobic amino acids, polar and charged amino acids. Okay, moving forward from here, so okay, we'll be discussing about the features or relationship between the contact order, long range order, folding rate. Okay, give me a second. So let's start discussing with the contact order. So contact order refers to the average sequence separation between the residues that form a non-local contacts in the folded state of a protein. Proteins with the low contact order tend to fold more rapidly than the proteins with the high contact order. This is because low contact order proteins have more compact and stable native state, which requires fewer long range interactions to form during folding. So here states that low contact order tends for few long range interactions as well. Coming to the long range order, the long range order refers to the degree of correlation between amino acid interactions in the protein. So protein with mistake here, low long range order tend to fold more. Coming to the relationship between the contact order is complex, and the two factors can be positively correlated many times. However, there are exceptions to these general trends. Also, the correlation between them is not so high. Okay, moving to the other uh, important one is multiple contact index, which is a measure of number of needle-like contacts that are formed during the folding process of a protein. It is calculated by comparing the contacts formed in a given conformation or with those formed in the native state of a protein. In general, proteins that fold via two-state mechanism tend to have higher MCI value than proteins via three-state mechanism. This is because two-state folding proteins tend to have more cooperative folding process where the formation of one native-like structure helps to stabilize the conformation of other contacts, leading to overall a higher MCI value. So overall, the MCI could be, there are exceptions to this general trend between the relationships of MCI and folding mechanism as well. So where the combination of contact order, long range order and MCI can be used in machine learning along with machine learning methods for predicting the folding rate. Moving forward, how this folding rate or folding is varied around different structural classes. So proteins can be broadly classified into four major structural classes based on the secondary structure, that is all alpha, all beta, alpha beta and alpha plus beta. So all alpha proteins have predominance of alpha helices in their secondary structures, whereas all beta have predominance of beta strands. Coming to alpha beta proteins, contain both alpha helices and beta strands, and alpha plus beta have regions of alpha helices and beta strands that are not contiguous. So in general, all alpha proteins tend to fold faster than other structural classes, likely due to their relative simple folding pathway and presence of stable alpha helices that can form early in the folding process. Additionally, all alpha proteins often have small size and a compact globular structure which may further facilitate the faster folding. Coming to all beta proteins on the other hand tend to have more complex folding pathways, often involving in the formation of partially folded intermediate states before reaching the folded state. As a result, they generally fold more slowly than alpha proteins, all alpha proteins. However, the folding rates of all beta proteins can vary widely depending on size, topology, and sequence properties. Alpha beta and alpha plus beta proteins typically have an intermediate folding rates that fall between those of all alpha and all beta proteins. The folding rates of these proteins can depend on the variety of factors including the size, shape of the protein, or the presence of disulfide bonds and the other structural features. So overall, while there are general trends in folding rates of different protein structural classes, there is significant variation within each class, and individual proteins can exhibit unique folding kinetic as well. So understanding the folding rate and mechanism of different protein structural classes can provide insights into the fundamental principles of protein folding and can aid in development of new methods for predicting the and designing the protein structures. Okay, here now I'll be starting with the protein interactions. So protein interactions are the interactions that occur between different proteins or between proteins and other molecules such as nucleic acids, lipids, or small molecules. These interactions are critical for many biological processes including signal transduction, enzyme catalysis, and gene regulation. So I'll discuss some types of protein interactions. First, I'll be starting with the protein-protein interactions. They occur between two or more proteins and they can be stable or transient. Stable protein-protein interactions are those that persist over long periods of time and are necessary for structural stability of multi-protein complexes such as ribosome or DNA polymerase. Coming to the transient protein-protein interactions, on the other hand, are those that occur between the proteins only briefly or often involved in the signaling or regulatory pathways. Examples of protein-protein interactions include interactions between the signaling proteins of RAF and MEK in the MAP kinase pathway, or the interaction between the transcription factors in the regulation of gene expression. Okay, moving with the protein nucleic acid interactions. Protein nucleic acid interactions occur between proteins and nucleic acids such as DNA or RNA and are critical for many biological processes including DNA replication, transcription, and translation. These interactions can be either sequence specific, where the protein recognizes and binds to the specific sequence of nucleotides, or non-specific, where the protein binds to the nucleotides in a more general manner. Examples of protein nucleotides interaction include interactions between the DNA binding protein, LAC repressor, and the LAC operator in the sequence in the bacterial gene regulation, and the interaction between the ribosome and mRNA during translation. Here is the protein-lipid interactions. Uh, these are important for many cellular processes, including membrane trafficking, membrane signaling, and membrane structure formation. So these interactions can be either electrostatic or hydrophobic in nature and may involve specific lipid head groups or fatty acid chain. Examples of protein lipid interactions include interactions between the GPCR receptor, rhodopsin, and the lipid bilayer in the retina, and the interaction between the lipid anchored protein caveolin and the cholesterol rich fat lipids in the plasma membrane. Next is the protein small molecule interactions. Structurally, small protein interactions involve incorporation of small molecules into the protein structure, such as binding of heme to hemoglobin for oxygen transport. So, in summary, these protein interactions are essential for many biological processes and can occur between proteins and nucleic acid, lipids, or small molecules. Understanding the different types of protein interactions and their characteristics can provide insights into the mechanism of these processes and may lead to the development of new therapeutics and other trends in biotechnologies. So here again I have some few questions, starting with the first one. A protein, which structure determines its ability to interact with the other proteins or molecules? Okay, yeah, 3D structure, it's correct. Tertiary or 3D structure or higher levels other than the tertiary structure helps. Moving forward to the second one, the interaction between the two proteins is typically mediated by specific. After this, I have only two more slides where we'll be moving for the today's actual problem solving session. Okay, any other answer for the second question other than the amino acid? It's a binding sites. Okay, so the specific binding sites help for binding of the other proteins. So they have the binding sites, or we call binding interfaces. Moving on to the third one, proteins that bind to DNA and regulate gene expression are known as. Can we DNA binding protein? Can you be more specific in the term? Can it be transcription factors also, right? Okay, transcription factors are also part of DNA binding proteins. Even that is correct. Yeah, here I asked particular class of a proteins which bind to DNA and regulate the gene expression. Next is the process by which protein loses its shape and function uh, due to histones don't regulate the expressions, right? So due to the exposure to extreme conditions such as heat or pH is called. Yes. Any other answers for the fourth one? Okay, yeah, it's denaturation. Moving forward for the fifth one, a protein's what determines its affinity for a particular ligand? So based on what factor or what we consider in a protein to determine its affinity for a particular ligand? So what on target exactly mean? Okay, it's a binding site again. So the active site, yeah, binding site. So moving forward for the sixth one, proteins can interact with each other through physical contacts such as, this is protein contact. Who answer? Okay, yeah. Hydrogen bonds and van der Waals interactions. Till now we have discussed about the importance of binding site in protein interactions. Let's come to know how we predict. Okay, here I'll be discussing mostly based on protein ligand interactions, but the similar thing works for the other types of interactions as well. So we have several computational methods that have been developed to predict the binding sites of proteins. So they include distance-based methods, accessible surface area-based methods, and energy-based methods. So we'll discuss one by one here. So distance-based methods predict the binding site based on the distance between the atoms or residues of a protein molecule and its binding partner. So these methods identify the regions of protein surface that are within a certain distance from ligand or other binding partners. For example, the pocket finder algorithm identifies the potential binding sites based on the presence of cavity or a pocket in the protein surface that is large enough to accommodate a protein. Next is access. Okay, briefly telling about the distance-based methods. When you have a complex, so consider the residues of a protein that are within a distance cutoff to its binding partner. Next is the accessible surface area. So this is based on solvent accessible surface area of the protein molecule. So these methods identify the regions of the surface that have high solvent accessible surface area and thus more likely to interact with the ligand. So in this solvent accessible method, so when we consider individual protein in its unbound form, residues will have high solvent accessibility. When we consider in the bound form, you have low solvent accessibility to them. So based on this phenomena, so residues which show high solvent accessibility in unbound form and which show high solvent accessibility in the bound form are tend to be the binding site, plausible binding sites. Next is the energy based methods. These methods use computational docking or molecular dynamic simulations to calculate the energy of interaction between the protein and ligand or its partner and identify the region of protein surface that have favorable binding energy. Coming, each of these methods has its strength and weakness, and the choice of the method depends on the protein interaction system that we used. Distance-based methods are generally faster and simpler, but may miss binding sites that are not in the cavity or pocket. Coming to surface area, accessible surface area based methods, they provide a more comprehensive view of the protein surface, but may not accurately predict the binding affinity of a ligand. And energy-based methods are more accurate, but are computationally more determining and may require high-quality protein structures. Yeah, researchers can choose from distance-based or accessible surface area-based or energy-based methods or a combination depending on the specific research question and availability of computational resources. Okay, moving forward for the propensity. So the binding propensity of amino acids varies depending on the type of interactions, and some amino acids are more commonly found in binding sites than others. Moving for the propensity of amino acids in the protein-protein interactions, now here we see hydrophobic amino acids such as leucine, isoleucine, and valine are commonly found in protein-protein interfaces, as they can form hydrophobic interactions with the hydrophobic residues. And aromatic amino acids such as phenylalanine, tryptophan, and tyrosine are also frequently found in protein-protein interfaces, as they can form pi-pi stacking interactions and hydrogen bonds. Apart from this, charged amino acids can also be present, are important for protein-protein interactions, as they can form salt bridges and electrostatic interactions. Moving forward, for protein DNA interactions, basic amino acids such as lysine and arginine are commonly found in DNA binding proteins, as they can form electrostatic interactions with negatively charged phosphate backbone of DNA. And aromatic amino acids such as tryptophan and tyrosine can also be important for protein DNA interactions, as they can form pi-pi stacking interactions with the DNA base pairs. Next is the protein ligand interactions. Now, aromatic amino acids such as tryptophan and tyrosine can be important for protein ligand and binding, as they can form pi stacking interactions with the ligand. And hydrophobic amino acids are also important, as they can form hydrophobic interactions with the ligand molecule. And polar amino acids such as serine, threonine, and asparagine can form hydrogen bonds with ligand, while charged amino acids such as lysine, arginine, glutamate can form salt bridges and electrostatics with them. Overall, the binding propensity of amino acids in different types of protein interactions depend on complexity of interplay of chemical, structural, and functional factors. Understanding the binding preferences of amino acids can help to design more effective drugs and therapies that target specific protein ligand interactions. Okay, here we are done with the summary part. So, if any doubts, I'll take up the questions. In a minute, we'll be moving forward solving the question. We have 8 or 9 questions for today. We can finish it off. Okay, for the first question in this sample solving session for today is, protein folding rates provide information on which of the following? We are doing the first question in the problem solving session. Protein folding rates provide the information on what? Yeah, here you have to be careful considering the question. In the question, it is given that protein folding rate. So, rate here gives how fast or slow the protein folding process will happen. So, protein folding tells us about the 3D structure from sequence. And why not option B? Because it is also tell fast and slow. Folding rate never tells about the unfolding things. Okay, we have studied about KF and KU as well, right? So here particular the question is KF, and option B here stands for KU. So it's the option is B. Protein folding rate provides information on how fast or how slow it assumes protein folding is done. Okay. Next is usually which types of proteins fold faster. I think I have discussed this and it is easy to answer. Okay, we are doing the second question. Which type of proteins, like which structural class of proteins fold faster? Yeah, I'll wait for a minute to see other answers as well. Okay. Yeah, moving forward to the solution, it's al- alpha proteins. It is generally true that all alpha proteins tend to fold faster than other structural classes such as alpha beta or alpha all beta. This is because alpha helices are relatively simple structural motifs and composed of repeating hydrogen bonds and stabilizing hydrophobic interactions between the amino acid side chains. This simplicity allows for a relatively faster and efficient folding of the process as the protein can quickly form its secondary structure and begin to pack its hydrophobic core. In contrast, okay, why not others? What they are lacking? All beta or alpha beta proteins often require more complex folding pathways as the beta strands must form hydrogen bonds with another before they can be packed together into a stable beta sheet. So this additional complexity can slow down the folding and increase the likelihood of misfolding or aggregation. But here still we have to note a point that not all alpha proteins fold quickly and there are many factors that influence the folding rate of a given protein. They include size, complexity, presence of disordered regions or either intrinsically disordered regions or post-translational modifications. Okay, moving to the third. What is the correlation between the contact order and the long range order? Okay, I think I didn't explain this. I'll be explaining about Chern plot. You guys can answer. I'll explain it. So this is a graph used to analyze the. Okay, I'll wait before getting answers from you. Then I'll give a explanation of Chevron plot. Okay. Yeah, it is used to analyze the folding kinetics of a protein. It is named after the chevron shape of a plot, which resembles the symbol for arrow, chevron arrow. So the plot shows the rate of folding or it could be unfolding as a function of denaturing concentration, usually using urea quinidine chloride as denaturing. The folding rate is measured as the rate constant for the transition from unfolded to folded state, and folding rate is measured as the rate constant for reverse transition from folded to unfolded state. The folding and unfolding rate constant are plotted on y-axis and denaturation concentration is plotted on the x-axis. Typically, the data is presented as a series of curves with each curve representing a different denaturing concentration. The curves show a sigmoidal shape with rapid increase in folding rate. So when you see this sigmoidal curve, you see rapid increase in folding rate at low denaturing concentration, followed by gradual increase in folding rate at high concentration. The point where folding and unfolding rate constants intersect on the plane represents the mid of the folding transition, where the folded and unfolded states are in equilibrium, that is CM. This point is also known as folding and unfolding transition state. The slope of the curve on either side of the transition state indicates the cooperativity of the folding process, with steeper slopes indicating the greater cooperativity. So these plots are useful for understanding the thermodynamics and kinetics of protein folding and can provide insights into the stability and folding mechanism of the protein. They are often used in protein engineering and drug design to optimize the protein folding and stability of the protein. Okay, let's move on to the sixth question. In an energy-based approach, which interactions are considered to identify the binding site residues in protein-protein complex? Yeah, one clue I can give here is, in an energy-based approach binding site, it starts with the long-range interactions. Okay. So energy-based methods, no, they'll be using algorithms to search for the potential binding sites based on the distribution of intermolecular interactions. So their goal is to identify the most energetically favorable binding site for the ligand, which is typically the site with the highest overall score. They have some scoring mechanism as well. So the first one, the answer is C. So in the preferential order, they consider van der Waals, electrostatic, and then hydrogen bonds. So these van der Waals interactions, we know, nonpolar atoms or molecules that arise due to fluctuations in electron density. So in context of protein interactions, van der Waals interactions can occur between nonpolar amino acid side chains and nonpolar regions of its binding partner. So these are the first preferential one energy-based approach it considers. Second is the electrostatic. Now we know these are attractive or repulsive forces between the charged particles, such as positively charged amino acid side chains and negatively charged amino acid side chains of the protein as well as its binding partner. So here answer is C. Even the D is correct, but preferentially van der Waals and electrostatic, then followed by hydrogen bonds. So moving to the seventh one, which database contains binding affinity of protein-protein complexes upon mutation? Yeah, it's proximate, which prothom contains the thermodynamic mutational data. The folding data is, is given in proximate. PDB sum is the other database that shows the interactions between the known protein complexes from the PDB. ProNit is for protein nucleic acid interaction thermodynamic database. So the correct answer is proximate. Moving forward, for binding site residues, PDB param provides the information on yes, it is distance between residue pairs. Okay, this is the last question for today. So if energy contribution from the main chain is not specific, while contributions from the side chain are specific for binding purpose or for recognition purpose, now I'm done with the questions and lectures today. So if you have any doubts, you can ask me. You can mention the chat box. If you are having no doubts, you are free to leave the session. Thank you for attending. See you next Monday. If you have doubts, please stay back. Mention your question in the chat box or unmute yourself and ask. If no questions, you are free to leave.
Sir, can you explain about Chau-Plasman method? Like it is going to be started from 10 power minus 4 only, na? The unfolding rate.
Please, can you repeat the question? Can you repeat the question, please? Yeah, okay. Someone is asking about question 8. So PDB param, I'm audible now. Yeah, PDB param, it has a, we have discussed about the different methods of which binding sites has been, like how to get a binding site. So we started with a distance based method, solvent accessibility based method, and then energy based method. So PDB param is a tool developed from our lab, so which it has several, so it gives us several structural information. So in this structural information, one such information is binding site residues. So in this, if you give a protein A and protein B, like as a complex or as individual, so it will give what are the possible binding site residue pairs in that. It's similar like a PDB sum. It's an alternative to PDB sum with an enhanced analysis and improved performance we have developed it. So when we consider this binding site residues option in PDB param, it won't show hydrogen bonds, it won't show ion bonds, it won't show disulfide bonds. Instead, all these interactions between protein are summarized as residue pairs in that. So this PDB param is a tool. Yeah, yeah, okay. I'll be talking about this correlation. So generally, when we talk about long range order and contact order, so proteins, if we take a stable protein, so it has a high long range order value and low contact order value. So similarly, the folding also, high long range order means it can easily form like the long ranger, the long range interactions, so which facilitate the faster folding it can form the stable secondary structures very fast in the early folding process. So this long range order represents the early formation of stable secondary structures in the early folding process, whereas contact order gives the compactness of the structure, overall compactness. So that's how one is high and one is low, giving them a negative correlation between them. Okay. So this chevron plot, it's what just a plot that plots between unfolding kinetics and folding kinetics, where this intersection point, you see this is transition concentration, like equilibrium concentration. The slope of them gives the what do you mean? Sorry. Okay, this plot shows the rate of protein folding and unfolding as a function of denaturing concentration. Usually, urea or guanidine is used as a denaturant. The folding rate is measured as the rate constant for transition from unfolded to folded state, and folded state is measured as the rate constant for reverse transition, that is from folded to unfolded state. So if you see x-axis we have denaturing concentration, and y-axis we have rate constant for folding or unfolding rate constants. So if you see these 2 points, these experimental lines have intersect at one point. So that point is called as a midfold of folding transition, where these states are in equilibrium here. So likewise, using this, we can get the Kf value, Ku value, where we can calculate delta Gf also from this. So these midpoints of transition, of slope of transition, are helpful for us in calculating the thermodynamics of folding and unfolding from this plot. Okay. I think this is discussed very detailedly by Sir in class as well. I don't exactly remember the lecture, but it has been discussed as well. Yeah, for folding, in this method, the calculation will start from folded structure, we add denaturant concentration, so on, and we get. Yeah, sorry. Yeah, you are right. For folding thing, we start from unfolded, go on. So you consider the reverse way here.